With each movement we do, action we take, sensation we get, our bodies combine the work of our brain, which sends and receives information, with our different tissues which act upon the brain's requests. Whether the action is looking around, running, or even involuntary, such as your stomach's digestion to food after eating, all of these are examples of the perfect brain-tissue collaboration. Inside the brain, ions move in and out of neurons in many different ways. One of those ways are the sodium-potassium pumps. Each sodium-potassium pump includes 3 Na+ sites and 2 K+ sites. The sites are highly selective and they do not allow any other type of ions to take a seat there. In a continuous cycle, one ATP molecule attacks the pump as its open site faces the inside of the neuron. ADP is released and the phosphate is affixed to the pump. 3 Na+ ions take place in their seats. The pump turns around and liberates the sodium ions just to hold 2 potassium ions instead. The phosphate group is released and the pump turns around again and accordingly transfers the potassium ions from the extracellular to intracellular space. The cycle goes on all over again. Note that these pumps work against the concentration gradients of the ions. It is the continuous work of these pumps that takes out a larger number of positive ions than that taken in, and thus keeps the intracellular space negatively charged with respect to the outside, as is seen in all recordings. The K+ concentration is always much higher inside the neuron than outside of it. In fact, any additional K+ concentration in the outside medium can heavily damage the nerves and therefore the nervous system's activity. The blood-brain barrier protects the brain from additional K+ ions coming in the blood in order to preserve its function. However, peripheral nerves are not protected and they might be damaged by excess potassium and by a lot of other ways. Tissue engineering targets cardiovascular system, integumental system, musculoskeletal system, and others, but also one of the very important tissue engineering targets are peripheral nerves. According to Paik-Zunzang et al., current repair methods for peripheral nerves include artificial synthetic material, which accelerates the repair process, seed cells which promote the formation of myelin sheath to protect and isolate the neuron, and peripheral nerve assistive technology, which enhances speed and accuracy of the axon regeneration. A major, very important system of our bodies that is a combination of the tissue and brain work is the visual system. Our visual system is very powerful and very smart. One of the many proofs of its amazing capabilities is what we call the visual adaptation. The visual adaptation is the process by which the visual system alters its operating properties in response to changes in their environment. These constant adjustments are very analytical and result in a lot of interesting sensations. In addition to the common example that visual adaptation is what allows us to read different handwritings, another beautiful example is presented by Clifford et al. Adaptation is in fact what allows us to know how someone is feeling from their facial expressions, for all different types of faces, for women, for men, for cartoons, and even for emojis. A second example presented by Webster et al. Is the following. If you look at the rose in the middle, you remark that it's clear, while if you look at the same exact rose in this picture, you remark that it's blurry. The view of the rose is affected by the environment around it. Cornea is one of the main targets of tissue engineering. Cornea protects the eye from dust and germs. It reflects light and allows us to see. How are new corneas engineered? First, cells from oral cavity are taken out by surgery. They are grown on dishes with feeder layer cells. These cells are not able to divide and thus they secrete growth factors that help the stem cells divide instead. After 2 weeks, we remove the layer by cell sheet process, which helps remove the cells together instead of collecting each one alone. Finally, we will attach the relatively transparent layer sheet to the eye without the need of surgery.